Okay, so let's talk about how sugar is made now. So yesterday we talked about how the first part, the light reactions, give you your ATP and give you your NADPH. Today we're going to talk about the Calvin cycle. Okay. Now this Calvin cycle is simplified, so you're not seeing all of the actual steps, but these are the steps that you need to know or worry about. Okay. So you can see basically what is going on in the Calvin cycle. We can see that there's a carbon input. So right up here, this is the carbon that's coming from where? Where is this carbon coming from? So you can see it right over here. Where's that carbon coming from? Coming from the, where? So the NADPH, I'm going to ask you a question. The NADPH and the ATP we talked about yesterday, that's made in where? Stage, sorry, not stage 1, but the light reactions, right? The hydrogen, I don't know if you can see that, but the hydrogen in the NADPH, what molecule does it come from? What molecule is split to produce hydrogen? Water. Very good. So, if you think about sugar, right? Sugar has 3 elements in it. There's carbon, there's hydrogen, and there's oxygen, right? This, the carbon, this comes from the water. Okay? Sorry, not the carbon. I just said that. I apologize. Not the carbon. Sorry. The hydrogen comes from the water. Okay? These two, carbon and oxygen, come from the environment, the air, or the water. Okay? So if it's an aquatic plant in the water, carbon dioxide is being pulled out from the water. If it's an aerial plant living in the air, the carbon dioxide is pulled out of the air. Okay? Now, the carbon dioxide is incorporated into the Calvin cycle by this enzyme right here, Rubisco. So let me zoom in a little bit so you can see it. Okay? So here's Rubisco. This is the enzyme right over here. This is the enzyme that incorporates that carbon dioxide. So this is what the Calvin cycle looks like, okay? What it does is this enzyme takes one of these molecules, it's called ribulose bisphosphate, and you can see that it has 1, 2, 3, 4, 5 carbon atoms. And what it does is Rubisco will add a molecule of carbon dioxide to this. Now, if we add one more carbon to this molecule, how many carbon atoms would it have? 6. So now you have a 6 carbon molecule, with 1, 2 phosphates attached to it. That molecule is highly unstable, and it will split in half. It will split in half to form 2, 3 carbon molecules. Where have you seen that before? A highly unstable 6 carbon 2 phosphate molecule splitting, Emily? Yeah, remember fructose 1,6 bisphosphate? Right in glycolysis, it splits into 2 molecules of G3P. Same idea. Okay? So what happens then is this will split and give you 3 phosphoglycerate. Now, if you have 3 of these molecules and they split, then you get 6 of them. So you get 3 phosphoglycerate. You can see the phosphoglycerate receives another phosphate. So we're using up energy here. So now we have a molecule of 1,3-bisphosphoglycerate because it has picked up an extra phosphate. This molecule gets oxidized or reduced. What's happening to NADPH? Is it becoming oxidized or reduced? Is it gaining or losing hydrogen? It's losing. So this molecule is being oxidized, which means that this molecule over here is being what? Right. This is being reduced. So the 1,3-bisphosphoglycerate ends up becoming G3P. Now, where have you seen this before? Where have you seen 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate? It's in glycolysis, but it's in the reverse order. In glycolysis, after the fructose splits, you get G3P, and then the G3P becomes 1,3-bisphosphoglycerate, producing NADH, which then becomes 3-phosphoglycerate. So I know that photosynthesis is not the reverse of cellular respiration. It's not, right? It doesn't look the same. It looks very different. But we can see some similarities, and we can see some things that are almost like the mirror image, right? So these three molecules are just like in the reverse order of glycolysis. So what happens is you have 6 molecules of G3P. You pull one out, you're left with 5, and those 5 G3P become converted back into RUBP. So it's a cycle because what are you getting back? You're getting back the reactants. Now, this kind of looks like the Krebs cycle, right? And in the sense that in the Krebs cycle, you get back a reactant too. What's the name of the reactant you get back in the Krebs cycle? Not acetyl-CoA. It starts with an OX also, which doesn't stand for oxidation. It's oxaloacetate, okay? So you get back your reactant. Now, we can compare this to the Krebs cycle. So in the Krebs cycle, do you generate or do you use up ATP? What happens? Do you generate or do you use up ATP? You make ATP, right? Because the whole point of respiration is to make energy. What about here? Here we're actually using up ATP here because we make ADP, and we're using up ATP over here. So we're actually, it's the opposite. We're using up ATP. In the Krebs cycle, do you make or do you use up NADH? You make NADH. What about here? You use it up. So in other words, the components of the cycle are being reduced, whereas in the Krebs cycle, it's the opposite. The components are being oxidized. Calvin cycle, do you consume, use up, or do you produce carbon dioxide? Calvin cycle, you use up, right? We're using up carbon dioxide. Whereas in the Krebs cycle, what do? You produce carbon dioxide, okay? Now, another name for the Calvin cycle is the C3 pathway. Why do you think it's called the C3 pathway? What do you think the C3 stands for? 3-carbon molecule. Because what's the first product that you get? You get a 3-carbon molecule, right? So it's also known as a C3 pathway. Okay. So that's the part that makes the sugar. Once you get that extra G3P, then what you can do is you can use the G3P to make things like sugar. Like how would we make glucose from G3P? How many G3Ps would we need to make glucose? Considering that G3P has 3 carbon atoms, we would need 2 of these, okay? So this is the part that takes in the carbon dioxide, uses up the ATP, uses up the NADPH, and converts that into sugar. So if you look at sugar, we have the 3 parts, the carbon, the hydrogen, and the oxygen. The carbon and the oxygen come from the environment, the CO2, and the hydrogen comes from the water that we split in stage 1. I don't know why I call stage 1, but the light reactions. Okay, so that's the second part. That's the synthesis of photosynthesis. All right, questions? All right. Now, I want to show you something that's quite interesting in regards to photosynthesis. I want to talk about something called photorespiration. Now, so we talked about photosynthesis, right? We talked about the two parts of photosynthesis. We talked about how light is used. We talked about how the Calvin cycle works. Now, when everything is working well, plants should be able to make sugar. Now remember, plants don't move. They don't move locations, right? They can move, but they can't really move from one spot to another. Okay? So they're not unlike animals. Animals evolve to eat other things. Plants evolve to eat the sun, basically. Okay? So their strategy is to stay in one spot, capture as much sunlight as possible, and that's what they do. Okay? And when all is going well, plants make their own food using the chloroplasts, and then they use the mitochondria to take all that food and convert it into what? Into energy. Okay? But sometimes things don't go so well. And this here, photorespiration, is when things don't go well. Okay? Because ribisco is an enzyme that's supposed to take in what molecule? CO2. Unfortunately, it has an active site that can also fit oxygen. So ribisco is supposed to take in carbon dioxide, but what happens is when the temperatures go up, it changes the protein in such a way that the protein actually starts to favor oxygen. Now, do you guys know your relative concentrations of oxygen and CO2? So the environment is what percent oxygen and what percent CO2? You guys know? What's the most abundant gas in the atmosphere? What do you think it is? The most abundant gas in the atmosphere. Because the air is a combination of gases, okay? The number one gas in the atmosphere, or the air, is nitrogen. The second most is oxygen. It's 20%, roughly, okay? Carbon dioxide is a measly 0.04%. I think it's 0.04%. It's less than 1%. Very small number. Even though the concentration of oxygen is way more than carbon dioxide, ribisco tends to favor CO2 because that's its job. Its job is to bring in CO2. But what happens though is when the temperature goes up, it tends to favor oxygen, and that's a big problem. That's when you get something called photorespiration. So I want you guys to do some math, okay? If I have a 5-carbon fragment, and I add one more carbon to it, how many carbon atoms do I have? 6. But if I have a 5-carbon fragment, and I take in oxygen instead, how many carbon atoms are in oxygen? 0. So what happens when I take in oxygen? Now my 5-carbon fragment does not become a 6-carbon fragment. What happens is it actually becomes a 3 and 2-carbon fragment, and that 2-carbon fragment, called glycolate, gets degraded, and when it gets degraded, CO2 is released. Now think about this for a second. Does a plant want to release that CO2, or does a plant want to take it in to make sugar? The plant wants to take it in and make sugar. So this is a huge problem. If a plant cannot take in carbon dioxide, it can't make food. It's in big trouble. So photorespiration is a huge problem. Now, and by the way, virtually all, there are some examples of plants that you don't use photosynthesis, okay? There's not many examples. But even like something like the Venus flytrap has to make its own food, okay? You're like, what do you mean? I've seen Venus flytraps eat flies and stuff like that. But the reason why it eats those things is not to make food, It's to get nitrogen. Because it lives in an environment where it cannot get enough nitrogen. Okay? But Venus flytrap can eat all the flies it wants in the world. But if it can't make its own sugar, it will die. Okay? It needs to make sugar. So, what's the solution? So here is, on the left, is a leaf of a typical, what's called C3 plant. Now C3 plant, all plants are C3 plants. What I mean by that is because if they're doing this, if all plants are doing this, which is the Calvin cycle, what's the first product in the Calvin cycle? A 3-carbon molecule. So all plants are C3 plants, okay? But some plants have added something on to that C3 part. They become called C4 plants or CAM plants. I'm going to explain that in a second, okay? So do you notice that the leaf on the left and the leaf on the right are not exactly the same? They look a little bit different. So you can see up here that these guys, these are cells. See how they're all like tightly packed up here? So this is the top of the leaf. And that makes sense because where's most of the sunlight going to be? The top. So here are your chloroplasts. And they're densely packed here because that's where most of the sun is, right? And remember, what do chloroplasts need to make energy? They need the sun. Now, a C4 plant does something a little bit different. You can see it looks a little bit different than this. So, what do they do? To deal with this problem of photorespiration, a C4 plant does something before the Calvin cycle. So here's your Calvin cycle over here. Okay, this is what we just looked at. Okay, here is the Calvin cycle. Here we have the carbon dioxide being brought in. But remember, photorespiration, the problem is Rubisco is not taking in carbon dioxide. What's it taking in? Oxygen. So what's the solution? By the way, what do we call that when an enzyme has two things that are trying to get into the active site? It's called competitive inhibition. Okay? How do you solve the problem of competitive inhibition? One thing you can do is to raise the concentration of the thing that you want. So if you have X and Y that are competing and you want X, you can increase the concentration of X to ensure that X wins. So this is what plants have done. C4 plants, that is. Okay? So here's your carbon dioxide. What happens, and you don't have to memorize this. You just have to understand the concept, okay? A C4 plant takes the carbon dioxide and adds it to this molecule, which is PEP, which is also a component of glycolysis. This molecule is a 3-carbon molecule. So what happens if you take a 3-carbon molecule and a 1-carbon molecule is you get a 4-carbon molecule. So why is a C4 plant called a C4 plant? Because before the Calvin cycle, they make 4-carbon molecules. What they do with those 4-carbon molecules is, so these are all 4-carbon molecules. So malate is a 4-carbon molecule. And you might recognize these are all components of what? Yeah, this is glycolysis, this is the Krebs cycle, this is the Krebs cycle, this is glycolysis. So you've seen these molecules before, okay? But the big idea is this. You take a 4-carbon molecule and you start to accumulate them. You start making lots of them. And then what you do is you take these 4-carbon molecules and you move them into the cell that's going to make the sugar. You take those 4-carbon molecules and you convert them back into 3-carbon molecules. You release all of that carbon dioxide. And what happens then is the level of carbon dioxide in this cell goes up significantly, way higher than it is in the atmosphere. Now the reason why you're doing this is because you have a problem, because you also have oxygen, which is in this case, my evil guy, and you want to make sure that carbon dioxide wins. So what do? you increase the amount of CO2 using this concept. You're basically using competitive inhibition to knock out the oxygen. So here's a question. Why don't all plants do this? Because this solves that problem. If plants can do this, they don't have to worry about the problem of not making sugar. But why don't all plants do this? So let me show you a slide, but you can actually, you don't really need to see the next slide, because you can kind of see it from here, but I'll show it to you anyways here. A C4 plant spends 30 molecules of ATP to make sugar. Yeah, C4 plant. A C3 plant only spends 18. So we may? Yeah, it comes at a cost. It's not free, okay? It comes at a cost. So the reason why all plants don't do this is because photorespiration is a problem when the temperature goes up. So if the plant lives in a cool environment, like many parts of Canada, you don't need to be a C4 plant. But if you're living in a warmer environment, like as you go south, then you might need to be a C4 plant. To me, the analogy I give often to my students, imagine it's like a fur coat. It comes at a cost, right? You got to pay for a fur coat or some sort of a heavy jacket. It comes at a cost, not cheap. When would you want to have a jacket? When it's cold, right? So imagine it's like freezing outside. I throw you in the wilderness and you don't have a coat. Now someone else spent a lot of money and bought a beautiful coat. So that person spent more money, but they're going to survive. You're in big trouble if you don't have a fur coat. But if I throw you in the desert and you bring that fur coat or the heavy coat with you, and you spend a lot of money on it, who's actually having an advantage there? It's the person who does not have a coat. So it depends on the environment. Okay? So if it's a warmer environment, C4 plant does better. If it's a cooler environment, C3 plant does better because it doesn't spend all of that extra effort making sugar. What if it's a really hot environment? What plant is this? Cactus. Now, another strategy to deal with photorespiration is the CAM strategy. Okay? Now, the CAM strategy is the same strategy as what I just told you. So I'm not going to go over it again. The idea is simple. The idea is you take your carbon dioxide and you store it in 4 carbon fragments. And then you take those 4 carbon fragments and then you break them up. You release all the carbon dioxide. And then you make sure that the carbon dioxide outcompetes the oxygen so you get sugar. Okay? The only difference is this. A C4 plant makes its 4 carbon fragments in one cell during the day, and then moves those 4 carbon fragments during the day into the cell that's going to make the sugar. A CAM plant does something a little bit different. It makes its 4 carbon fragments at night, and then during the day, it takes those 4 carbon fragments and releases them. Now, why? Because where is it living? Where do you think this thing lives? In the desert. It's really hot, and there's also not a lot of what? Not a lot of water. Trivia question. How does a plant get gas in and out? Through what feature in the leaf? What is it? Not the cell wall, it's right here. You know what these things are called? Yeah, the stoma. Okay? Now these are little openings. You can see it here. Here's one. Here's another one. If you're living in a really hot environment and you open up at night, you open up those stomata during the day. So the problem is when you open them up during the day and it's really hot, what can come out? Yeah, moisture. It's like sweating, right? It's like opening up your sweat glands. Same idea. So what they do is they close the stomata during the day. They close them. Okay? They open them up at night. Why at night? Because it's cool. So if they close their stomata during the day, they don't lose as much moisture. If they open them during the day, it's really hot, they're in big trouble. Because yeah, they'll be able to get gas in and out, but then water is going to come out too, and then they'll dehydrate, and that's not good. So the CAM strategy is exactly the same as the C4, just the difference is they do the carbon storing at night, and then they do the carbon releasing during the day. That's the only difference. Okay? So here's a question for you. Why didn't, this seems kind of like pretty elaborate, right? Like here, I'm going to ask you guys a question. Imagine you made something, right? Like, we talked about the enzyme that runs the Calvin cycle, and you can see there's a big defect in it, right? There's a huge defect in this enzyme. This enzyme is supposed to take in carbon dioxide, but sometimes it screws up. There's a big flaw that when the temperature is warm, it doesn't take in carbon dioxide. And if it doesn't take in carbon dioxide, the plant will die. So the solution is to do all of this craziness. Look at it. Look at this. This is pure and utter craziness. Why couldn't we just get a better enzyme? Why? Why couldn't we just make a better enzyme that doesn't take in doesn't take in oxygen? Why not? It's an interesting question. Very interesting question. So another interesting question is, this tells you a lot about when Rubisco actually evolved. When it evolved, the environment was different than it is today. What do you think it was different? I think it was different. When Rubisco first appeared, when it first evolved in photosynthetic organisms, what do you think the concentration of carbon dioxide and oxygen would have been? Was that? The opposite. Very good. It would have been very high levels of CO2, very low levels of oxygen, and the problem of photorespiration wouldn't have showed up because you would have very little oxygen to show the defects. It wasn't until many, many years later, and I don't know how many, I'm assuming millions and millions and millions of years later, because what does photosynthesis make? It's oxygen, right? As the oxygen levels start to go up, then this defect started to show up. The problem is, life is not like a car. If you have a defect with your car, you can take it to the mechanic and you can get it fixed. Life doesn't work that way. If there's a defect, you can't do the same thing you would do with the car. Because to take the engine out of a car and fix it, you can't do that with a living organism. What would happen if you take something out of a living organism? It dies. So you can't exactly do that. What you have to do is come up with a different way. Although it looks like a crazy way, you have a defective engine. You can't really change the engine, so you got to come up with a way to solve the problem without really changing the engine too much. So Rubisco evolved when the environment was really different. Carbon dioxide levels were really high, and oxygen levels were really low, and this defect didn't really show up. Okay? Okay, so listen, we're almost done. We're almost done. The last thing I want to show you is some graphs in regards to photosynthesis, because it's a neat way to review a lot in this whole unit, because this whole unit, really the main idea of this whole unit is enzymes and how enzymes work, right? How they're affected. So I thought I would end up with showing you some graphs because it has to do with enzymes. Even though we're talking about photosynthesis, it has to do with enzymes a lot. So here's a graph that shows you light intensity. So this is on the x-axis. This is light intensity, how bright it is, and this is how quickly we take in carbon dioxide. Okay? So this is your photosynthetic rates, basically. Because remember, what does photosynthesis take in? Carbon dioxide. Take a look at this graph. Where have you seen this pattern before? It goes up, and then it levels off. Where have you seen that before? Right. Reaction rate versus substrate concentration. It's exactly the same pattern. So what do you think the explanation for this is? As it gets brighter, the plants take in more carbon dioxide. And that makes sense because if it's brighter, there's more what? There's more light. If there's more light, there's more energy. If there's more energy, then we can run photosynthesis faster. And to take in carbon dioxide, we need to have enough what? ATP, and we need to have enough NADPH. And where do those come from? The first part, the light reactions. So if that increases, the rate increases, right? As we get brighter, it goes up, but then why does it level off? Why doesn't it just keep going up? What was the explanation in the reaction rate versus substrate concentration? Excellent. Because there comes a point where the enzymes cannot go any, what? Faster. There's a theoretical limit, right, to how fast the enzymes can go. Okay? What's going on in this graph? This graph looks like it's the same. See the exact same pattern. Here's your brightness. Goes up, levels off. Because the enzymes can't go any faster. But we can shift it up if we increase the temperature because the molecules are moving faster. Right? We know the temperature affects the rate of reaction. So these are the good ways to review enzymes, right? And how they work and how their performance is affected. Where have you seen this pattern before? This is reaction rate versus what? Temperature, right? Remember how it goes up and then it comes down? And it goes up because what's happening on this side? Here, the molecules are moving what? Faster. And then we get to some sort of an optimal point. And then here, what happens? As it gets too hot, the enzymes start to change shape, and they don't fit their substrate very well. So even though the molecules are moving more quickly, the effective collisions drop, and the rate goes down. But now, I want you to think of another explanation we can talk about too. If it's a C3 plant, what's another possibility? Photorespiration, right? As the temperature goes up, the rate goes down because of also photorespiration. Okay? This is temperature on the x-axis. Okay. This is temperature here. This is your photosynthetic rate over here. Which plant does better? Depends. So if it's cooler, like 20 degrees, you go here, take a look. At 20 degrees, the C4 plant is pretty stable, but the C3 plant makes more sugar. So what plant does better? C3. But at 35 degrees, the C3 plant makes less sugar because of photorespiration, but the C4 plant makes more sugar. Okay? What about at, what plant would you want to be at about 27 degrees? Charlotte, if you had to be a plant, which one would you be? At 27 degrees. C3 or C4? So they make the same amount of sugar, right? But I think I would rather still be the C3 plant because the C3 plant does not have to spend as much to make the sugar. And And then there's this one. So what do you think this is a graph of? What are you seeing here? As we increase the amount of what? Oxygen, the rate goes down. So this is what? Competitive. This is competitive inhibition. Okay? So ladies, that is it for Unit 1. We covered enzymes and energy, we did biochemistry, we did cellular respiration, and we did photosynthesis. Your test is only going to be on respiration and photosynthesis though, okay? It's not going to be on the first chapter. Alright ladies, so that is the end.